The ABTA is pleased to welcome you back to our webinar series. My name is Umbreen Mann, Program Manager here at the ABTA. Our webinar today is Clinical Trials, Paving the Way Forward. Thank you to today's sponsors, SDP Oncology and Orbis Therapeutics. Today's webinar will be given in two parts. We will begin with an overview of clinical trials and how they pertain to adult brain tumors, after which we will delve into clinical trials for pediatric brain tumors. Stick around till the end to hear from our patient speaker who will share insights from his firsthand experience with the clinical trial process. Without further ado, I'm delighted to introduce our first speaker for today. Dr. Michael Vogelbaum is Program Leader of Neuro-Oncology and Chief of Neurosurgery at Moffitt Cancer Center and Professor of Oncological Sciences at the University of South Florida Mersani Medical School. In addition to his active neurosurgery and radiosurgery practice, he has been primary investigator of numerous local and national clinical trials of new drugs, surgical techniques, and devices for brain tumors. Thank you so much for joining us, Dr. Vogelbaum. You may now begin your presentation.
Thank you, Umbreen, and it's really a pleasure to be speaking on behalf of the American Brain Tumor Association and helping to discuss the important role that clinical trials play in the treatment of patients with brain tumors. Next slide, please. In terms of disclosures, this of course is always very important whenever we give talks. And you can see some of the companies that I work with, and this is really all in support of clinical trials and drug and device development, which is essential for us to be able to move forward. And I'll talk about that a little bit. Go ahead, next slide, please. And my talk is really focusing us initially on just the general concept of why are we talking about clinical trials. I'll be focusing on clinical trials for adults with brain tumors, and Dr. Packer will be focusing on clinical trials for pediatric patients with brain tumors, but the basic principles remain the same. And so I'll help lead you through this question, why clinical trials? Next slide, please. So let's start with a definition. I think it's important to understand exactly what is a clinical trial, because there are other things that we do that are sometimes called clinical studies, and they're different. And so we want to really focus in on what a clinical trial is. And the National Institutes of Health is a federally funded agency that is very important in funding biomedical research in general, and also is important in funding many types of clinical trials. So they've defined clinical trials as a research study in which one or more human subjects, patients, are prospectively assigned to one or more interventions, which may include, in general sense, placebo. We'll talk about why that's not the case in cancer trials, or other controls, to evaluate the effects of those interventions on health-related biomedical or behavioral outcomes. The point here is that this is a designed intervention. It has a protocol that defines how it's going to be done, and that all of that work is done before the first patient is treated. Next slide, please. And that is important to make a distinction to other types of studies. So what are examples of clinical trials? Well, it could actually be a comparison between two established treatments to see which one is better. So there's nothing really investigational in terms of one treatment or the other. It's the investigation is the comparison. Or it could be evaluation of a new treatment or other intervention to determine whether it's useful for patient care. And most of our trials focus on that, see whether or not something is actually safe and effective. And that really points to the next one, which is an evaluation of whether a new type of treatment is safe and effective at helping patients live longer. And there are many different types of clinical trials. Some of them are listed here. Mechanistic trials, where we ask questions about how a new therapy works and whether it's likely to help benefit patients live longer. Some are early stage, exploratory or developmental. Some are trying to determine whether a new treatment is feasible. And then there are other types of interventions and even behavioral trials that evaluate the impact of a treatment on a patient's ability to function and their well-being. Next slide, please. So what is not a clinical trial? Sometimes it helps to understand what is a clinical trial by evaluating all the things that are not. In general, use of treatments that are available to physicians without special regulatory oversight and that are being used without the intention of collecting data or subsequent analysis. We're not doing, we have information that's going into the electronic medical record, but there's no special effort to collect additional information. And so for brain tumors, many of the things that we do routinely, we do outside of a clinical trial, including surgery, a biopsy or use of lid or surgical resection, radiation therapy, radiosurgery, tumor treating fields, FDA approved medical therapies, such as chemotherapies or targeted therapies or immunotherapies. And the asterisk there is to note that some of those therapies are not actually approved for brain tumors, but we as physicians are allowed to use any approved therapy within certain constraints, certain limits. But we often will use therapies for other cancers to try to treat patients with brain tumors when it makes sense. What's interesting is that imaging, as I'll discuss later, is also subject to regulatory oversight, but there are plenty of imaging modalities that are approved for patients with brain tumors, including MRI, CT, and PET imaging. Now, there are many times we will look back. We'll look back at a group of patients who are already treated, not on a clinical trial. We'll look back and say, how did they do? What were the factors that could have predicted how someone might have responded? What can we learn from that experience? When we look backwards and then go to the electronic medical record or other sources to try to gather that information together and do analysis, we call that a clinical study. That's not a clinical trial because the treatment was already done. Now we're looking back as opposed to designing an intervention in the setting of a clinical trial. Next slide, please. So as I mentioned, there are times that an approved therapy can become part of a clinical trial. And even, and there are certain examples here where we could take therapies that we use routinely, but subject them to a clinical trial. For example, a study that randomizes patients with a brain metastasis to surgery or radiosurgery to ask the question, which therapy is better for that patient under certain conditions? Or a study that randomizes patients with an oligodendroglioma to radiation plus one type of chemotherapy versus radiation plus a different type of chemotherapy. That's actually an ongoing trial, what we call a phase 3 trial, and I'll explain what that means. That's an ongoing trial, an international trial that's still enrolling patients. Or a study that compares one dose of radiation therapy to another. We have quote unquote standard doses we often use for treating patients, but those are not written in stone. We arrived at those doses because of some clinical studies and clinical trials, but sometimes we try to optimize things further. And a study that evaluates the use of radiosurgery to treat recurrent pituitary tumors. We do all these kinds of studies prospectively sometimes to be able to address a question in the most careful manner. We get the highest quality information or data back from those studies so that we can make the best possible interpretations and decisions about where to go next. And really the point here is that all of these examples have in common that there's an intent to collect data prospectively in real time to evaluate the success or impact of a prescribed treatment. Next slide, please. Now, there are certain circumstances where we cannot do a treatment without doing a clinical trial. And this is really, I think, what's most relevant for patients with malignant brain tumors, as we'll talk about later. There are, as I mentioned, some treatments that are given as standard of care therapies. These are ones that are generally accepted to be effective. But others can only be given in a clinical trial. And as an example, those are treatments that have not been approved by the FDA for any diagnosis or indication. For example, new therapeutic devices or even ones that accompany a therapy, delivery devices, for example. New drugs, new types of chemotherapy or targeted therapies or biologics, things like gene therapy or antibodies and cellular therapies, We've heard about CAR T, that's a cellular therapy. These may never have been approved for use in any cancer, and certainly not in brain tumors. They have to be subjected to a clinical trial in order to be used. And again, it's to figure out whether or not they're safe and how to use them most safely, and then go on to determine whether or not they're effective. This also applies to imaging. For example, if there's a new imaging modality or new contrast agents or tracers, these need to be subjected to a clinical trial. Interestingly, when a drug is approved for another cancer, for example, and it's given by a pill, by the oral route or an IV, and instead we want to try to put it directly into the brain, even though it's an approved therapy, what is part of that approval is the route of administration. So when we take an approved drug and change the way it's being given to a patient, that is now an investigational treatment. Because there's a whole new host of risks that we need to understand in order to be able to use it most effectively and safely. Next slide, please. And the point here is that clinical trials require special oversight. There was a time when there were new interventions that were being tested in patients without any oversight and without any consent. We refer to that as the bad old days. These things happened, unfortunately. People were hurt very badly. There was a large uproar. There were times that certain vulnerable groups were subjected to these quote unquote trials, which were not really trials. They were reckless. And it's because of that we really have to do this in a robust regulatory environment. And the reasons are, for some of these new therapies, the risks to patients may not yet be known, and we need to understand that. Some kinds of new therapies are very complex and require special training, and this needs to be documented and understood and shown to be effective. There are ethical concerns about randomizing patients to one treatment versus another. I know that a lot of patients often feel uncomfortable that a computer is making the choice, and sometimes don't recognize that when a physician makes the choice, it may be no different from the computer making the choice, but that is an ethical problem that needs to be addressed. And ultimately, patients are going into trials because they're hoping for benefit in a situation where we don't have cures. And so we feel like there's a moral obligation for us to be able to collect and analyze the data in the most rigorous manner. Patients are investing themselves in this process. We need to do the same. And so clinical trials have the highest degree of rigor. Next slide, please. There are special processes that are involved in doing this. So as I mentioned, for new treatments, the FDA has to grant permission for a trial to be done. That is the purpose of the FDA, is to make sure it's being done well. And this involves an extensive submission of preclinical, what we refer to as laboratory or animal study data, showing that a drug or device or a new therapy is likely to be safe and effective. And then there's an extensive review of the planned clinical trial protocol. And what is a protocol? It's a very long document, usually over 100 pages, that details every aspect of what is going to be done, how it's going to be monitored, how safety is going to be evaluated, how problems will be reported, what interventions will need to be done if there's a problem. The whole thing is documented in a protocol, and that needs to be reviewed. In addition to the FDA review, there has to be an evaluation by a local board, an institutional review board at each study hospital. And many hospitals, especially what we call comprehensive cancer centers, like the one I'm at, have an independent scientific review panel as well that also looks at it. So there's several different layers of review, which seems onerous, but it's important. It helps, again, to preserve safety and make sure that we're doing the clinical trial in the best possible way. There are all sorts of formalized data collection, auditing, and monitoring processes. And on top of that, there's a separate consenting process. When I do surgery on a patient, I talk to them about why we're doing the surgery, what the alternatives are, what the risks are, what the potential benefits are. and then they sign a consent memorializing that discussion. When it comes to a clinical trial, that's not enough. It actually has to be a much longer written consent document that lays that out in writing, and then the patient can sign that. Next slide, please. So we often talk about clinical trial phases, and I just want to talk about this a little bit so that, again, we're on the same page in terms of what we're talking about, and this will also be important for Dr. Packard's talk. So we start with a phase 1 study, and this may be a first in human use of a new type of therapy, or the first time in a specific population of patients. So if it's the first time being used in a human, of course, that makes sense. We're going to start actually with a relatively low dose and start marching our way up in terms of the dose that's given to the point where it's not tolerable. And that's often the case with old drugs, chemotherapies, some of the new treatments. We actually do not see as much in the way of harmful side effects, and we go up to the highest feasible dose. But the reason we also do this in a setting where we have a drug that's already on the market, and now we're going to be evaluating it in patients with brain tumors, is because there may be interactions with other drugs that only the brain tumor patients are getting, things like steroids or anti-seizure agents. And we need to make sure that the drug is still safe in that population of patients. So we really get that safety profile. We're also looking to see, though, that it's effective. Now, one of the areas that I've been focusing on is also asking this question of whether or not the drug gets to brain tumor tissue in an effective manner when we're also looking at the safety. And we call that a window of opportunity trial. There's another term that's been used, which is called phase 0. Doesn't completely apply to brain tumor patients, but it's really asking the question, what is the drug doing to the tumor? And that's part of that initial evaluation. Why do we do that? Well, we have not always been doing it. We really should be doing it more. And that's because most drugs given orally or IV, given systemically, as we say, can't get into the brain very effectively. We have a blood-brain barrier that keeps them out. And so we need to understand whether or not the drug is actually getting there, because what we don't want to do is start giving lots of patients this new drug that never actually got into the brain. We really want to avoid that. And that's the whole point of doing these kinds of window opportunity trials, and that's part of the phase 1 process. Next slide, please. So phase 2, now we've established what is the highest dose that we can give safely. And now we ask, what is that initial efficacy? Are we starting to see some clinical effect in patients? And we compare that to a control population. And that control, as we say, is it may be prior patients treated with standard of care. And we're asking, are the patients being treated with the new drug doing better than them? Or sometimes it's a randomized phase 2 where we're actually treating some patients with the current standard therapy and some with the investigational therapy on top of that or instead of that, and asking the question, do they do better? And it's a small population. This is not really enough to get to the point of full approval usually, but the point is to start saying, are we seeing some signal there? Can we identify subsets of patients who might really benefit from this new treatment? Now, why do we do this? Well, in the end, any kind of clinical trial, especially when we talk about phase 3, which we'll get to next, any type of approval process is reliant upon statistics. So what is the point of that? Well, what we're asking is, what is the smallest population of patients from which we can make a prediction about all the others? So let's suppose that there is 100,000 patients out there with a certain type of brain tumor, and we want to know whether or not a new drug is likely to be effective. Well, we can't test that drug in all 100,000 patients. They won't do it. Most of them won't elect to be in a clinical trial, but it would also be logistically impossible. So what we want to ask is, if we were to look at 100 patients, okay, can we project that result out to the next 100,000? And that relies on all sorts of statistical techniques. And usually the answer in the brain tumor world, when we want to ask, is something truly effective, it usually takes somewhere between 500 and 1,000 patients. Well, that's a lot of patients. It takes a number of years to actually treat those patients before we get an answer. And we want something earlier to say, is this one likely enough to be effective that it makes sense to then expand to that 500 to 1,000 patient population? And that's what we do in phase 2, essentially. We're trying to hopefully see a fairly strong signal and see what patients might benefit the most. So that's the point of a phase 2. Next slide, please. And then the phase 3 is ultimately the gold standard, so to speak. And that's where we really make that final determination. Is this going to be effective, and can it become standard of care? And this is always going to have a comparison to standard of care. Patients will be randomized. Usually it's 1-to-1, meaning for every patient who gets the investigational therapy, there's going to be another who gets the standard of care. But sometimes, and more recently, we've been doing different types of randomizations. For example, 2 get the investigational treatment and 1 gets the control or standard treatment, and sometimes as high as 3. Now, there's an important point to be made here. Even when we use a placebo, okay, that does not mean no treatment. What it means is maybe no treatment beyond the standard of care, but there is always going to be standard of care treatment provided in any cancer trial. And we're asking the question, is the investigational treatment really better than on top of standard care? Is it really better than standard of care alone? And as much as it always gives people pause, it is an important principle, because as I'll point out later, most of our therapies that we put in clinical trials so far have not shown efficacy, and some have actually added risk. And so we need to ask that question. We're always thinking optimistically. We want this new treatment to work. We feel that way, patients feel that way, but we really need to understand, because we really don't want to create more harm. And there is that risk. So that's why we have to do this in a randomized way. And so that's the whole point of the blinding, is that we know actually from lots of trials in the past that when people think that they're getting the investigational treatment, sometimes they think they're better, everybody thinks they're better, but when we look at things that are not subject to that kind of opinion, that is how long someone lives, sometimes it shows that they really weren't any better. And so when someone knows what they're getting, sometimes it can change the interpretations, at least in the short term, but not necessarily in the long term. So it's important to understand that, and that's part of that process of blinding. Next slide, please. So now that we've gone through the different types of trials and what we talk about, and there are a few other variations on the theme that I did not discuss, things like pilot trials and the like. But now that we've talked about these in general terms, I'd like to talk a bit about where do clinical trials come from? And I'm not sure that people fully understand where these come from. And so really, in the end, they start with a question, okay? Is there a better way to treat patients with brain tumors? That's what we're all asking. And then it comes to more specific questions. Is this new therapy I've been working on, is that safe? Are there certain patients in which it might be effective? How can we improve on the ability of new or existing therapies to fight brain tumors? And those are trials that we often do with established therapies, and adding new things and taking away things. Are there combinations of treatments that work better? And if so, how are they used together? That's where all trials start, with questions like that. Next slide, please. And ultimately, it takes a clinical trial team that has to work together to try to obtain answers to these questions, okay? And I've listed some of the members of the team: physicians, scientists, nurses, coordinators, data managers, statisticians, industry. And I've highlighted physicians in industry. that is often a danger zone for us in the academic world to say that we're working with industry. We cannot advance care in any disease, much less brain tumors, unless we partner together. What we do have to do is maintain some firewalls so that we don't have inappropriate conflicts of interest, but we cannot advance the field unless we work together. And that goes on both sides. And why is that? Well, physicians, we understand the disease, we treat the patients, we have that relationship that is central to our professional lives. Industry, they have the technologies, they have the resources, the money to be able to develop new treatments. Sometimes those new treatments actually come out of what we call academia or universities. They're physicians and scientists working together who come up with new ideas, and then industry takes them along the commercialization process so that they can actually go into patients. I'm not sure if anyone really understands how much it costs to develop a new drug, but before a new drug has ever gone into a patient, industry has already spent tens of millions of dollars. That's what it takes. And not even NIH can pay all of those costs. So it's very important that we work together. And then there are other important members of the team, like the nurses, the research nurses in particular, who help to take care of patients on a clinical trial. The coordinators who make sure the trial protocol is being followed and enable us to really effectively follow that protocol. Data managers, as I told you, there's a high degree of rigor, and we have to collect the data in a very careful way so that we're absolutely certain that we're collecting good data. They're essential. And then, as I described, the statisticians are key to this because they're the ones that help us to determine how likely it is that the treatment we're working on and the results that we get can be extended to a larger population. And as I said, physicians alone or together with industry design and run individual trials. Plenty of trials are purely investigator or physician run. Modifications to existing therapies or evaluations of new combinations of approved therapies. We've all written and run those kinds of trials. But almost everything else we do, especially when we're talking about developing a new therapy, that's physicians and industry working together. Next slide, please. So there are many sources for these new trials. Sometimes it's industry that's developing a therapy who comes to the physicians and say, hey, we have this great new drug. We think it might be active in patients with brain tumors. They put together the protocol, they write the study design, and then they look for physicians and hospitals that will be willing to put patients on the trial. And those often involve multiple hospitals. And then there are ones where the investigators or the physicians come up with the idea, develop the trial, and then go to the industry, the companies that have the drugs that are involved, and say, hey, we have this great idea for your drug. We really think it's going to work in our patients. Will you be willing to support this trial with drug or with drug and funding? Because trials cost a lot of money to do. And that's an investigator-initiated, industry-sponsored trial. Next, please. Next slide. Thank you. And then there are trials that are actually funded by the federal government. So the National Cancer Institute is part of the National Institutes of Health, and they have a budget that is in part used to support clinical trials that are designed, developed, and run by physician scientists. So these can be investigator-initiated trials that are done at one or multiple hospitals. We have what used to be called cooperative groups, or lots of hospitals, hundreds of hospitals working together to run innovative clinical trials. The current term is the National Clinical Trials Network, NCTN. And what's important about understanding these trials is that there is an incredibly rigorous peer review process. That is, and I've done this many times, when we put together a trial idea and what's called a concept, a trial concept, or a protocol, we have to go to a panel of other physician investigators like ourselves, and we have to convince them that this trial is important to run. And often that requires a discussion and some modifications. They actually add to it. They actually add some ideas, try to make it better. And what comes out is hopefully the most scientifically rigorous trial that we can run. And then we obtain the funding from the federal government to run that trial. There also are philanthropically funded trials, which are often investigator-initiated trials, and they, again, may be single hospital or a network of hospitals. And then brain tumor centers like ours, we develop individual trials all the time. Things that are available to our patients at our site, usually early ideas. We have a new idea, we want to start trying it and see how it works. And if it actually works out well, then we start partnering with other hospitals and going to those larger scale trials. And often these are funded by philanthropy or grant funding. Next slide, please. Now, this often comes up, and I think it's an important point that we have to address. Who pays for all of this? Because in the end, someone has to pay for it. There's a lot of work involved, a lot of people, and there has to be some mechanism to pay for it. And as I've mentioned, clinical trials most often involve a mix of standard of care, which we would normally do without a clinical trial, and then there's an investigational component, that which we're doing on top of the standard of care. It used to be that insurance companies would say, okay, you propose a surgery or radiation therapy for a patient with a brain tumor, fine, we pay for that, that's normal. But now, oh, wait a minute, you're proposing surgery and radiation therapy and you're adding something to it. Well, we're going to pay for nothing. It's now a clinical trial. We don't pay for research. And of course, there was still that standard therapy component of it. Now they're saying they're not going to pay for any of that. Well, that was put to rest in 1995 when Medicare said that's just not right. Still have to treat the patients. So even the standard of care treatments at that point, they mandated that be paid for. But there were still times when that applied to Medicare, the Medicare program, and some insurance companies followed it, but most did not. And then in 2010, as a part of the Obamacare legislation, it was mandated that all insurance companies have to at least pay for the standard of care component. They don't have to pay for the investigational component. That's something that is paid for by the sponsor, so to speak, either the drug company or device company or the federal grant that would help pay for the investigational part, but the standard of care had to be paid for as if it were being done off trial, which is only fair. And so that's the way it pretty much works today. Any aspect of a clinical trial that is not the standard of care has to be paid for by the sponsor, including extra MRIs, for example, extra diagnostic studies, blood draws, any other costs that are associated with running the clinical trial. All those people I talked about, the study nurses, the data managers, the statisticians, and so on. That has to be paid and by the sponsor, and that's only fair. Next slide, please. Now, there are a number of challenges to running clinical trials. We have to specify, as I told you, that there's a lot of rigor, there's a lot of oversight. We have to be very strict. There are strict eligibility criteria. We have to define who are the patients who we are planning to treat. And that involves a lot of detail, age, type of tumor, timing, whether it's newly diagnosed or recurrent, certain laboratory values to make sure that we're staying within a safe range. And all of that becomes written in stone. Small deviations, can, no matter how minor, are usually not tolerated. So, and it comes down, and this is part of the experience of learning how to be effective at writing a clinical trial. If we say, okay, the platelet count has to be quote unquote normal, all right, and that's one of the criteria for it. And if the platelet count, let's say the upper limit of normal is 450,000, and the platelet count is 451,000. That patient is no longer is not eligible any longer. Okay, even though that's a minor deviation that is of no clinical consequence, they can't be. So we've gotten smart about writing these things in ways that matter because we don't care about it being 451. What we care about is whether it's below the lower limit of normal, and especially much lower than the lower limit of normal, because then there's a risk of bleeding. And so we're getting smarter about writing these things so that we can try to make more people eligible. Often treatment can be done only at the hospital that's offering the trial. This is a real problem. This is a real challenge, especially for newly diagnosed patients. So let's suppose that someone is diagnosed with a glioblastoma. They went 100 miles away for surgery, and then are offered a clinical trial at that hospital. And that clinical trial is being done with a new therapy that's being added on top of the conventional radiation and chemotherapy that's done for glioblastoma. Well, what it often means is that chemotherapy and radiation have to be done at that hospital 100 miles away. And when you're talking about daily treatment over 6 weeks, that can be very hard on people. Now, if that treatment is added after the chemotherapy or the radiation therapy is done, then it's often feasible. That's not as much of a problem. But if it's a new treatment that's being given during the radiation, it has to be done at that hospital. And that's because of the safety side of doing a clinical trial. We need to be able to assess the patient every day during treatment to make sure that there's not an unanticipated interaction that could be very harmful. And there's a certain amount of experience and rigor that will occur at the study hospital that may not occur at the community hospital down the street from the patient. And so that's why that's an issue. Not all trials have that limitation, but very often it is. And then there are strict timelines and limitations on prior treatments. And again, this is all part of the rigor of doing this. Next slide, please. Next slide, please. Thank you. So are there alternatives? Are there other ways for us to figure out whether a new therapy is safe and effective? Why do we engage in clinical trials when they're so hard to do? I've given you all the challenges here. And it turns out that less than 4% of oncology drugs evaluated in clinical trials make it to FDA approval. And nearly 80% of clinical trials fail to meet their enrollment projections. And the result is the process of taking a new drug candidate from first in human trial to approval can cost nearly $2.6 billion, okay? Now, some have taken that to say our clinical trial systems are failing, okay? If only 4% of these drugs can get through, it's failing. I would take a different view here. I would say that what our clinical trial system is doing is being very careful to make sure the only ones that get through are truly effective, that we're not using patients as guinea pigs, that we're not throwing things at them, hoping they don't get harmed, and then making claims about efficacy that cannot be supported. The fact that 96% of drugs don't make it through to approval means that we actually have a very, very robust system for generating new ideas, generating new drug candidates, and testing as many different things as we can in order to find that needle in the haystack, that one thing that's going to truly work. And if our clinical trial system were not effective, then there far, far fewer new drug candidates would be entering into it. It would be impossible. This is enough. 4% is enough to keep a very robust system working, and it really creates a lot of room for new ideas. Next slide, please. But there are alternatives that have been proposed. For example, a real-world approach. Okay, and that means using patient registries, so routinely collecting patient. As I said, when we look back, we call that a clinical study, but the idea of a registry is to actually look forward without calling it a clinical trial, and then also doing what's called data mining of electronic medical records. And the idea here is one can use this, compare one treatment option to another in a real world setting, and try to come up with a better way to treat patients, to optimize treatment. And there are some examples where that may actually work. And there's also the idea been raised that instead of doing clinical trials for new drugs, going through safety and then efficacy and so on, then just demonstrate safety and then just put it on the market, and then see how things go by using registries and electronic medical records. And a lot of these ideas have been battered around, and in fact, the FDA has taken them quite seriously. And this is actually a publication from their website about a report that they issued on this. Next slide, please.
Thank you, Dr. Vogelbaum. Just to let you know, you have about 2 more minutes for your section of your presentation.
Great, thank you. All right, so real-world evidence has its own challenges. These are unaudited data sets, and it's very hard to know whether or not these things are truly effective. And you actually have to have drugs that are approved to be able to do this. So it is being incorporated slowly, but it's going to be a process. Next slide, please. All right, so why consider a clinical trial? Next slide. So benign brain tumors, these are often surgically curable. Why do a clinical trial? Because surgery doesn't always cure these. And sometimes we can look at medications that may do a better job. This is an example of a response in patients with craniopharyngioma to a new drug. Next slide, please. But for malignant brain tumors, this is where it's really critical. Next slide. Take, for example, brain metastasis. Because of clinical trials, we have moved from a situation 3 decades ago where patients with brain metastasis were sent home to die, to the point where 2 decades ago, we now showed that surgery and radiosurgery were effective. And a decade ago was really more radiosurgery. And this occurred because of clinical trials, phase 3 trials. Next slide, please. And now we're at a point where we have some new drugs that look like they can show some efficacy, some targeted drugs, some immunotherapies. Next slide, please. And so is it time to stop using surgery and radiosurgery? Some medical oncologists are saying yes, but we don't really know yet. Only about 50% of patients respond to these new drugs. So we have to really do clinical trials to figure out whether or not these new treatments are truly effective in which patients, and whether or not combinations with the other therapies we have might be better. Next slide. But it's really for primary brain tumors where we really have to do clinical trials. We have some treatments that are effective, but they're not cures. Next slide. This is where we really need to develop new approaches. I have an example here, and you can look at the slides later, of grade 3 tumors where we learned by doing a clinical trial that we can actually take away some therapy, that really radiation alone followed by chemotherapy is better than radiochemotherapy followed by chemotherapy. Next slide. And that's what this example is here. So we can move on to the next slide. But for GBM, we have tried so many different treatments, so many different things, and they haven't worked so far. We have to get better at this. Next slide. This is only going to be clinical trials, and we have to start asking the questions of why have our treatments not been so effective? And there are lots of reasons shown here of why they may not be so effective, why our preclinical models may not work. Next slide, please. And so the approach then is to do what I was saying earlier, and that is window of opportunity trials, where we can actually ask these questions in patients. We can give the drugs, look at the tumor tissue, and see what's happening. And next. And ultimately, we can also deliver therapies directly into the tumors, directly into tumor-infiltrated brain. There are many different examples of this. I show an example of a trial that Dr. Packer may refer to in pediatric patients, where we can deliver therapies directly into brainstem gliomas. Next slide, please. Ultimately, I just wanted to highlight this one. And this is a trial where we're both delivering therapy directly into tumor-infiltrated brain, and also doing a window of opportunity component where we can ask what happened in the tumor tissue when we gave this very new type of therapeutic. And in this way, we're going to be able to understand, does this new treatment have biological plausibility? And then we can deliver it to the part of tumor that we leave behind, the infiltrative part that we cannot remove safely. That is ultimately where hopefully we're going to be going in the field. Next slide. So how do you find clinical trials? Well, at comprehensive brain tumor programs where you have highly coordinated care provided by a number of specialists. And sites like this have wide variety of different types of clinical trials that I talked about, not always close to home. Next slide, please. But we also all work together in these large clinical trial networks that I mentioned. Sometimes some of these trials can be closer to home. They're great sources of information from clinicaltrials.gov, the Brain Tumor Network, and of course the ABTA, which has always been a great source of information for patients. And I believe that's my next to last slide. If I can have the next one, please. And then in the end, how do you choose the right trial? This comes up all the time. There is no such thing as a curative investigational therapy. If it was curative, it wouldn't be investigational. So all of these are potentially curative or potentially effective, but you can't really say that one is better than another until it's gone through a clinical trial. And so ultimately, there's a relationship with the treatment team that's so important. Next slide. And it also is a matter of one's overall philosophical approach. Is this a sunrise or a sunset? Are we going to make progress? Looking at the picture, one could argue either way. Next. It's actually a sunrise that was taken over Tampa Bay. We all require big teams in order to do this kind of work. We require funders as well. And hopefully we can stop cancer in your head and hoping to deliver on that. Some license plates I've used in the past. Thank you very much.
Thank you so much, Dr. Vogelbaum. Our next speaker for today is Dr. Roger Packer. Dr. Packer is Senior Vice President of the Center for Neuroscience and Behavioral Medicine and is director of both the Gilbert Neurofibromatosis Institute and the Brain Tumor Institute of Children's National Hospital. Much of his research has focused on the development and performance of clinical trials for adults and children with neurologic, neuro-oncologic, and neurogenetic disorders. And he has received peer-reviewed grant support for his research. Thank you for joining us, Dr. Packer. You may now begin.
My pleasure. I wanted to thank the ABTA for offering me this opportunity, and we'll try to shorten it a little bit. Dr. Vogelbaum did such a great job in outlining so many things. For disclosures and support, I have no stock or patent rights, and I do have some consulting and advisory board agreements. Next, please. One of the concepts for pediatric brain tumors, and what I'm going to try to do, is try to put an emphasis on what's different about doing clinical trials for pediatrics. Is one of the major things is, unlike the situation in adults, where 3 or 4 different kinds of tumor types really dominate the landscape of tumors, in pediatrics, we're dealing with a lot of splinter tumors, tumors of different types that occur in especially children under 14 years of age, and with entities such as high-grade gliomas and low-grade gliomas occurring, but also entities such as embryonal tumors, medulloblastoma, other tumors making up a significant amount of the numbers of children that we have to treat and find solutions for. Next, please. For the older children, which fortunately have less brain tumors, more brain tumors occur in younger children. The tumors are more like the adults. We start seeing meningiomas on the astrocytomas, pituitary tumors, but it's really a different world in the younger patients. Another point to make out that even tumors that have the same names in pediatrics and adults, like high-grade gliomas, are different tumors molecularly. So the therapies that the adult groups use may not be applicable to pediatric tumors. Therapies that might work in adults may not work in pediatrics, but the corollary is there too, that some therapies that might not have worked in adults might be effective in pediatrics. Next slide, please. The other point just to bring out is that pediatric brain tumors don't only occur in the top of the brain, in the cortex. Over half of them occur below the top of the brain in an area called the posterior fossa, where we have the brainstem and the cerebellum. And especially these brainstem tumors can be extremely difficult to treat. And we'll talk about how we try to approach therapy for those tumors. Next, please. So basic issues with pediatric brain tumors include that clinical trials are considered standard of care in pediatric oncology. This is really based on work that was done in leukemia, showing that with these kind of studies, we can make dramatic improvements in survival, and no insurance company will deny the at least standard treatments that are required just because a child has gone on a study. They may deny the investigational component of the study, but standard of care is being on a pediatric study for pediatric neuro-oncology. Increasingly, because we know pediatric brain tumors are different biologically than those in adults and are different from one another in many cases, increasingly our studies are what are called translational, where we take advances in the laboratory and try to bring them quickly to the bedside. And many of our treatments that I'll show you are molecularly based, meaning we have to know the molecular genetics of the tumor before we can even start treatment. And this means that tissue is increasingly required. But the good news is that on very small amounts of tissue, because of the great advances in science over the past 10 years, we can do remarkable molecular analyses to give us a better insight in the tumor. Nuances between those studies in children and adults. The regulatory aspects that we'll talk about in pediatrics are more difficult, especially when we're talking about generalizable knowledge, the multiple different tumor types. The numbers available for a study are smaller. We're very careful to limit the number of so-called comparison or phase 3 studies we do because it will take years and years to complete. And by the time we complete them, some of the information will be out of date. And we have many diseases, unfortunately, where there is no acceptable standard of care. So there's no indication to do a phase 3 study. Next, please. We've already talked about this. Michael talked about this, and I'm not going to go over them, except to say that phase 1 and 2 studies are very frequent in pediatrics. Phase 3 studies are very specific for some tumor types like medulloblastoma, possibly ependymoma, and newly diagnosed low-grade gliomas. But otherwise, we don't have standards of care. And we want to increasingly, as it was discussed, try to go into the pre-treatment window of opportunity phase 0 studies, but we are limited in what we can do because of NIH and FDA regulations. Next, please. Where did these studies come from? That was mentioned, but in pediatrics, for parents, there's often a bewildering array of places they come from. For most centers are part of the Children's Oncology Group, over 100 centers in the United States and North America, largest, have multiple studies, a real focus on phase 2 and especially phase 3 studies. The second government-funded study group is the Pediatric Brain Tumor Consortium. We've been members of that for about 20 years. It's the NIH supergroup. Until recently, it was only in the United States. Now Toronto has been added. This is a group that is charged with doing cutting-edge technologies, intricate studies, but really are doing phase 1 and phase 2 studies and trying to transition their studies to the Children's Oncology Group. There's a Department of Defense Neurofibromatosis Clinical Trials Consortium. And in Europe, there's an International Society of Pediatric Oncology, which is Eurocentric, is international, but is really very much driven by individual countries that have banded together. Next, please. Increasingly, there are other groups that have been very active, non-NIH funded groups like the Pediatric Neuro-Oncology Consortium, Connect. These tend to be more industry friendly, more agile. They tend to fill a void so industry doesn't have to form its own studies, but can work with a preset group. We're a member of multiple groups like this, like PNOC and Connect. There are institution-led consortiums. There's a consortium out of Florida led by Dr. Mitchell and others that are focusing on immunologic problems. And there are occasional in pediatrics industry-run studies, but probably less than in adults. Single institution trials are great, and they're important, but because we have so few kids at any individual institution, we have to really think hard if that's the direction we want to go rather than banding together. Next, please. This is the pediatric research regulations, which is critical. And it codifies that to children, you cannot do more than minimal risk studies without direct benefit to the patient. If you're doing it for generalizable benefit only, just for knowledge, there has to be either minimal risk, a little bit more than minimal risk, or you can't do it. So we have to be smarter in how we do our studies. Next, please. This is the government rule about minimal risk. You can read it, but what it really says that if we do anything like an intervention, a biopsy, there has to be some benefit to the patient. And this is not the prerogative of the parent or the prerogative of us. These are the rules to try to protect children, and we have to understand that and be smarter as we get into our window of opportunity studies to do them appropriately so we can gather the critical information, but at the same time fit into this risk of not harming a child or taking the risk of a child who can really not consent to that part of the study. Next, please. So I'm going to give you a few very brief examples of how this fits into the real care of children. The most common childhood brain tumor malignant is the medulloblastoma, and it is an excellent example of how clinical trials in a stepwise fashion has improved survival. These have been well performed. They've been focusing on progression-free and overall survival, but they have not done as good a job as we want to really focus on some other things like functional outcome, and that's something we're going to rectify in the future. These tend to be long trials, and there's always the risk that they're not keeping up with the science. But increasingly, molecular stratification, because these groups get smaller and smaller, magnifies the issues, and we have to be very smart as we perform these studies. Next, please. This is what the tumor looks like, large tumor, back of the head. This is a side view, often with spread tumor. This is the medulloblastoma. Next, please. And this is looking at survival curves. And we're taking a look at these survival curves. You can see if the top is 100% survival and the years from study, over the decades, we have gone from that lower light blue curve up to that green curve by stepwise adding more effective therapies. Now, the counter of this is we get up to survival rates nearing 80, 85, 90%, similar to the leukemia studies. We start potentially doing therapies that are not only focused on survival, but increasing quality of life and maybe reducing therapy. And that takes the risk of lowering the likelihood of survival. And unfortunately, we just completed a study that we were all sure was going to be safe, but the less aggressive therapy was less effective. And without doing these trials, we never would have proven that. Next, please. We also have the understanding that we're dealing with pediatric patients with developing brains. And this is what is a scatter diagram. And on the left-hand side is where this child's IQ started before they started treatment. You can see because of the tumor itself, about 2/3 of our population had IQs less than 100. But unfortunately, after 2 to 3 years after completing treatment, many of the children lost 10 to 15 IQ points permanently, stressing why these randomized trials are not only trying to improve survival, but also improve quality of life. Next, please. And one other thing we're learning the hard way is that these trials may cause secondary tumors. Or could it be because the child was prone to develop the secondary tumor because of cancer predispositions? And now we know that over 1 in 10 children, or 1 in 10 child who has a brain tumor will have some predisposition in their germ cells and that will predispose to cancer, germline predispositions. And we may have to alter therapies for those children. Next, please. And the last part of the puzzle of medulloblastoma is not one disease. We know, looking at these children molecularly, that there's probably as many as 13 subgroups of medulloblastoma, which puts us in an extremely important sort of breaking point in our development of clinical trials. If we try to develop a clinical trial for every one of the 13 different subgroups, we'll never finish any of them. At the same time, if we don't understand the subgroups and move children into different arms or potentially different therapies, if we can lump them together, if we don't do that, we're going to fail in our new treatments, especially molecular targeted treatment. Next, please. The challenge is considerably different in low-grade gliomas. This is actually, when you tie them all together, the most common childhood brain tumor. We do have effective treatments, meaning that very few of our children with low-grade gliomas will succumb to disease. But functional outcome is really what we are after in many of these children. How do we prevent visual loss? How do we prevent seizures? How do we give them the best quality of life? How do we delay radiation if the radiation is going to hurt their brain development? And here is where dramatic new biologic insights over the past decade and clinical trial work working together hand in hand has made potentially the largest advance we've seen in brain tumors since we raised survival in medulloblastoma a couple of decades ago. And for the first time, we're looking at molecular targeted therapies that may allow us not only to avoid extensive surgeries for some patients and radiation, but actually chemotherapy. And we've gone from discovery to safety to effective therapy to randomized trials in less than 10 years in this patient population. Next, please. And this is, we know that some tumors like this large white tumor that you're seeing in the middle of the brain with chemotherapy, next slide, can shrink significantly. And we can show survival curves like on the next slide that show that we can control in children with neurofibromatosis 3/4 of patients with chemotherapy alone for many, many years, maybe forever. And even in those without neurofibromatosis and low-grade gliomas, in 2/3 we'll have some degree of control for up to 5 years. But then we start losing control in these patients. But it isn't that we're having ineffective therapy. We just don't have a therapy that works perfectly. And we also don't have a chemotherapy that improves quality of life. Next. However, the introduction of molecular therapy has really made a difference. This is a child who is nearly blind, age 6, with this very large tumor that was sitting on the child's chiasm. The child was considered legally blind for the past 6 months, and it failed chemotherapy and radiation. Next slide. After one cycle of a drug called bevacizumab, this tumor shrunk down to this, and this child actually regained vision. And it was a real teaching point to us that is, and this was done as part of a clinical trial, it is a real teaching point to us that we can regain neurologic function that we think in some cases has been gone for a long time if we use the right molecular targeted therapy. Next. And if you don't believe me, this is what is called the visual field on the child, where these are over time. And the dark area, as the child looked out of one eye, was the part of the world that was missing. There was a field cut. And then on this therapy, over about a 6-month period, the field cut got less. You could see the dark areas disappearing. And by the completion of therapy, the field cut was essentially gone. Unfortunately for this treatment, there's a great deal of rebound. You can't use the blood vessel drug for long periods of time. You have to stop after a year, and often the tumor came back. Next. But then the big development occurred in 2008, where there was a discovery that the majority of low-grade gliomas were truly a single pathway disease, meaning there was a single chain of molecular events because of a variety of different mutations, most commonly a mutation in something called the RAS MAP kinase signaling pathway in the gene BRAF, that allowed the tumor to grow. Next. And that we, luckily, because of industry and others, had a drug that was being targeted for melanoma, also that had an aberration in this RAS MAP kinase pathway. And we could get access with partners in industry, partners at NIH to the drug next. And we could see responses like this, baseline to 6 months later, next. And if we took all the children in one arm, and this is all children with low-grade BRAF mutated tumors, and we looked at how many of them responded, this is what is a waterfall plot showing the amount of growth or decrease in growth of the size of these tumors. And you can see all the ones to the right, the tumor shrunk anywhere between about 20% and 80%. And in only 4 out of the 25 did the tumor grow on treatment. Next. And in children with neurofibromatosis that also have a defect in that pathway, it was 100% successful in at least shrinking the tumor to some degree. Next. So we are now in a randomized phase 3 trial after completion of the phase 1 trial that showed it was safe, a phase 2 trial that demonstrated efficacy, to now see how it compares to standard chemotherapy in what we call a 2-to-1 randomization. We don't control who goes on one arm, comparing it in both children with neurofibromatosis, next slide, and children without. And this is the way that we need to go. Now, the thing we're going to build into this also to see if we have functional improvement in these children. Next, please. Unfortunately, the story is different with diffuse midline gliomas, or DIPGs, diffuse intrinsic pontine gliomas. We've made tremendous steps in our understanding of the biology based on getting tissue for the first time. For many years, these patients were treated without tissue. However, we made no or little progress in overall survival. Biopsy is becoming an increasingly important way to obtain tissue in these patients. We know that if we do the biopsy, molecular information is obtainable in over 90%. It can direct therapy, but it may not improve it yet. And here is where that common rule makes a big difference. Next, please. Because for this tumor, families are willing to say, if all I can do is radiation, at least biopsy it so I can help other generation of children. It might not help mine, but we can't do that. We have to, because of the idea that we cannot put harm to children who cannot assent for themselves. And for doing that, we have to develop trials that we are going to use the tissue to direct our therapies for that child to make it potentially better, and at the same time try to utilize this tissue to help other children and other generations make progress with this tumor that is 95% deadly within 18 months. Next, please. And this is the rule we talked about. If it's more than minimal risk, and biopsying a brainstem or biopsying a tumor is more than minimal risk, then we have to figure out a way to show that we are doing something good for the family, or something that we're using the tissue to direct our therapies before we can just do the biopsy. It isn't that we don't want to learn, and it isn't that we don't want to make progress. We just can't hurt children on the way in. And this is the balance we're talking about. And it's a very difficult one ethically and on many different levels, but it is what drives us all. Next, please. The last area where I think there's tremendous interest in pediatric brain tumors is immunotherapy. I've been around long enough that where immunotherapy was going to cure brain tumors in 1980, and it didn't exactly do it, but we're getting smarter. We know that it's been of tremendous help in different kinds of tumors, and it's the new, renewed great hope. We have many targets and many approaches, and thankfully not enough or not many children that were going to need this therapy yet. So we have to be very judicious in what therapy we try and let the science lead us which direction we're going to go. The problem we have is, unlike the information that we have for molecular targeted therapy, where we have a good way to at least assess where the target is the target there? Is a genetic defect there in the tumor? For immunotherapy, because of the lack of what we call humanized models, we use animal models that may not tell us how the human immune system will respond, or we use models where the models are immunosuppressed to allow the tumor to grow. Those models are not going to give us information. They are not going to be informative models. So we got to, in many ways, we're jumping from theory to therapy. In others, we need better humanized models. And we have to decide who is the best patient to treat. Is it the tumor, or is it the environment around the tumor that will determine if the therapy is effective? Are we going to get swelling when we do this because we're unleashing the immune system? Is the child going to be able to tolerate the brain swelling in that way? And how are we even going to analyze if something is working? If our therapies cause transient swelling, we might think it's not working where it really is. So that's called pseudoprogression. So those are extraordinarily important factors for immunotherapy that we want to push into our families and push towards it, because we think there are some children we've just hit a wall with, and we've got to go better and do things better next. And this has not stopped people from trying. These are the numbers of pediatric immunotherapy trials. Unfortunately, yet in pediatric brain tumors, we don't have one that has shown efficacy, but we're still very young in this rapid upswing of, since we've identified checkpoint inhibitors and other vaccines. So it's really an exciting time to care for these children. We have to understand that we need tissue sometimes to direct therapy, even for children with relapsed disease. We may need tissue because a tumor can mutate over time, and the therapy we targeted for newly diagnosed patients or a newly diagnosed tissue will not tell us the new mutation, and we may fail for that reason. And increasingly, we are recommending to families to biopsy recurrences, not to know what the tumor is, but to know if there's a different molecular change. And we're increasingly utilizing these molecular targeted therapies and immunotherapies to try to make a difference. What our major goal is to improve survival, but our secondary goal, which is extremely important, is quality of life. And as we go into these therapies, we have to be cognizant that we're dealing with a developing nervous system, a developing brain, and we may not know the long-term benefits of our therapies for 10, 20 more years of what they've done to brain development. But at the same time, the only way we're going to make progress and treat patients better for that patient and future patients are by clinical trials. And I'll just leave you with the idea, the right clinical trial gives you the best available therapy, plus something that could be potentially value added, and we hope relative minimal disease to the minimal risk to the patient. That's why you got to keep pushing. You got to talk to organizations like ABTA, Pediatric Brain Tumor Consortium, COG, institutions, and just see what's available and if it's worth the inconvenience to try to improve quality of life by getting the child on that clinical trial. And thank you for your attention.
Thank you so much, Dr. Packer. It is now my honor to introduce our patient speaker today, Chris Ferry. Chris is an ABTA volunteer, mentor, and strong advocate for the brain tumor community. Thank you so much for joining us, Chris. The floor is all yours.
Hi, can you hear me all right?
Yep, we can hear you.
All right, perfect. Well, I want to say thank you to the ABTA for having me on and thinking of me for this particular webinar, because I am a huge proponent for clinical trials. Very recently, on March 12th of this year, I officially completed my maintenance phases of the clinical trial of an immunotherapy drug that I was on. I was one of a few other people that were still on it and having shown very good response to this particular immunotherapy drug. And it's, you know, I hear, like, during the course of this discussion, we're talking about how, 96% of clinical trials fail. I don't really see it as that, because the data that we're gathering right now, or the data that we gathered 15 years ago, could be vital to the next treatment that will be a clinical trial 5 years from now. We're still gathering information on every single trial. So while they may not have done their exact purpose, we're still gaining information from them. And it's information that we can be using in the future and guiding our future decisions. Now, I want to, sorry, I had all of this all planned out in my head, but after being on, well, I had so here's a quick recap of what happened with me. I had my surgery, which was an emergency brain surgery. I only had 36 hours to prepare for that. And then my doctor, thankfully, at MGH, they were very proactive in trying to get me informed on multiple clinical trials that the hospital was offering. So I was diagnosed with a glioblastoma. I had my resection surgery, and I was offered both a option of doing the standard of care, which is your radiation and your temozolomide, or I could do the radiation and then, starting the same day as that, get infusions every 2 weeks and then every 4 weeks for this immunotherapy drug called durvalumab, which has been used in other cancers. And I happen to be qualified in that I had not been given bevacizumab or had done any prior standard of care treatments. So I had the choice, and I didn't have to choose that day. The thing that's really, I had no knowledge about clinical trials before any of this happened. So the first thing that I thought of was cost. I was like, how are we going to be able to afford to pay for this? And then when I had the follow-up meeting with my doctor, she let me know that all of the costs, as was covered in the webinar, any of the costs for that would be paid by the sponsor, and I didn't have to worry about any sort of additional cost. So I had the option to do the clinical trial, and if it didn't work out, I could always fall back onto the standard of care. So I always had the option to go back, and I still have that option to do the standard of care with temozolomide. But I'm really happy to say that it's been 4.5 years now, and my last MRI, there has been no recurrence, and my next scan is actually 1 week from today. Hopefully it will be the same thing. And if you are able to take part in a clinical trial, I would highly recommend it. You can search for them on the clinicaltrials.gov or the other resources like the ABTA or the Brain Tumor Institute, and try and, be proactive and talk with your doctor, because there are so many different like trials that are recruiting right now. I mean, granted, I'm lucky that I live in an area where I can go to a local hospital, and I know that's not a possibility for everybody. But try and be proactive in finding a clinical trial that suits your particular tumor, because what worked on me might not work on somebody else. And that's why we have to keep going, keep testing. And like the doctor said, there every tumor and brain cancer, it's not one single disease. And if you think about the mutations that each tumor can have within itself, it grows exponentially. And trying to find that one treatment that works for one person, that's why so many of them fail. But as we wrap up here, I just want to say thank you so much for having me on. And I want to thank the ABTA for being such a great resource for me for the past 5 years, because I went out and found them when I was looking for resources and information regarding my brain tumor diagnosis. Because like most people who have been diagnosed, we don't know anything about brain tumors or brain cancer when we're diagnosed. Like it's an entirely new experience for everybody. And I think that's about it. I want to say thank you so much for having me on, and it's been wonderful.
Thank you so much, Chris, for sharing your story and your experiences. It's really inspiring to hear what a positive experience you had and how well you're doing now. I would like to invite Dr. Packer and Dr. Vogelbaum to come back on as we'll take some questions. Just as a reminder, if you do have questions that you'd like to ask, please type and submit it using the question box in the webinar control panel, and we'll answer questions as time allows. So one of the first questions we have here, Chris, you talked about how you were lucky enough to live in a city where you had access to some great, well-renowned hospitals and brain tumor institutes. Unfortunately, not everybody lives in an area like that. Are any of you aware of resources that help provide financial assistance when it comes to traveling or finding stay when patients who are in more remote areas are having to travel for their treatments or clinical trials?
So Roger, do you want to go first? Because often that's more available in the pediatric world than the adult.
No, go ahead. No, go ahead. I'm going to be pretty unhelpful here.
Yeah. So in the adult world, some of the costs are for housing and lodging, right? So the American Cancer Society has had Hope Lodges. I believe they still do, and other things like that. Some clinical trial centers will have additional local community resources that they can point you to. I was thinking in the pediatric world, there's the Ronald McDonald houses, and then there are even institutions that provide free care and housing, like St. Jude in the pediatric world. But it's, traveling's hard. No matter, even if you take the cost away, there's a family support system that's so important for patients with any kind of cancer, and certainly with brain tumors. That we try to take that into account when we think about our trials and how to do them more effectively. And I can assure everyone who's watching this that there are active discussions of how to modify our clinical trial designs to be able to better incorporate community centers. Some of this is going to require changes at the level of the FDA, the National Cancer Institute, But that is something that we're all working on to try to make it easier for patients to be on clinical trials.
One of the more interesting byproducts of COVID, and nobody thinks very positively of COVID, is there have been regulations that have been loosened by the National Cancer Institute, by sponsoring agencies, allowing more of the tests to be done closer to home, not the actual infusions of drugs, but many of the tests that you have to do, the MRIs. And that's really helped some of our families. And I think that we are looking very hard with these agencies, whether this should be a permanent change. Agencies just like me change in a very glacier-like way. Sorry about that, but we really have to take some of these lessons learned. And as Michael said, McDonald's House, occasionally St. Jude's, if you have an open trial that you fit into, they will support. We try to build some inducements into some of our studies, but it's never enough, and it's just hard. But it's remarkable how mobile families are these days and figure out ways to get to different places. And then we have to figure out ways to change our schedules to make sure the MRI and the exam and the infusions on the same day so they can get in and out. And that's part of the game. And if we do our job, it's not as burdensome, I hope.
Thank you so much. Another question we have is asking, when a patient is offered a clinical trial, what are some questions or things that they should consider to ensure that they're making an informed decision?
Well, I guess, it's a lot of that is done in the process of the consenting and the like. I talked about the fact that there's a more extensive written consent that's involved with doing a clinical trial, because that's the goal, to try to give as much information as possible. And the biggest challenge is one of information overload. I think that asking about alternatives is very important, asking about what the schedule is going to look like, how many times they're going to have to come into the hospital for blood draws and other aspects of the trial. I think those are important things. In the end, the comparison of one trial to another, I mean, we get this question all the time: which one of these trials is better? Which one is going to be a better therapy? Well, I wouldn't be offering both at my institution if I thought one was truly better than the other. It's just they're each better alternatives, we think, than doing nothing, certainly, and hopefully better than standard of care.
The only other thing that I would add, and I completely agree with Michael, is that we could do a lot of overload with these trials. I think we really have to do a very good job to explain why a trial is better than standard therapy, why you would need to go on that trial. As far as comparisons, there may be 8 options. There may be only one option. But at the end of the day, you have to make a recommendation to that family when you're not fulfilling your job as a physician. You can lay out the different options, but you need to make a recommendation. At the same time, when we make these recommendations, you just got to be sure you're not putting tremendous pressure. And one ways an institution can do that is have an internal prioritization and say, look, there are 5 things, but because of timing or phase of the study, phase 2 versus a phase 1, we are recommending this. And this is going to be a prioritization for all patients with that diagnosis at that time period. And that takes out some of the biases. But there's no perfect way.
Thank you so much for that insight. I think we can take one more question, and this one is asking about the length of clinical trials. Do clinical trials always have an end date? And are you aware of maybe like a common amount of time that it takes a trial to go from being a clinical trial to then becoming standard of care?
I'll start with this one. It really depends. Where there is no standard of care, if you see a major improvement, it's going to be the standard of care. And my bias is that most things that work are going to hit you in the face. And it's not going to be 10 years to get a 5% increase in survival. I don't think that anybody wants to do those kind of trials. We're doing our best in pediatrics to move from trials that took 7 to 10 years to complete to no trial is going to be out there for more than 2 to 3 years without coming to some answers. That's the only thing fair to the science and to the patient. I don't think we're there yet. We're still opening trials that are going to take 7 to 10 years. It drives me crazy, maybe because I don't have 7 or 10 years left to see the end of that trial. That's neither here nor there, but also because it's just not fair to where the science is going. So we have to be smarter in trial design. And Michael talked about that. And, and yes, trials do have to have an endpoint.
So I'll just add another point to it. And so it's important to understand there is no one time at which a trial expires, because it also depends upon what you're studying, which disease. So for example, a glioblastoma clinical trial, those generally are over after a couple of years because we get an answer pretty much after a couple of years of whether or not it's extended survival. We're also doing trials in low-grade gliomas in certain subtypes where the expected survival is 15 years or longer. So when you start thinking about doing a trial to see if someone's going to live longer on a new therapy in a disease like that, you're looking now at 20 years, 25 years before you get an answer. I'm actually in the middle of one of those kinds of trials. I helped design it a little more than 10 years ago. And all of us who were involved in designing it knew that it was the trainees who were in the room who were going to end up being the ones who published the paper on the answer to that trial. We would be long retired before we have that answer. But it's also an important one to get because that's a situation where we want to make sure that not only people are living longer, but they're living better. And that's one of the key questions we're asking is the new therapy or which therapy is going to have less side effects and make people and help people live longer. So that's an important question to answer in that population of patients.
Breen, before we end, can I just say one thing quickly?
Sure.
Especially because Chris is on the line. This sounds like a homily that it's just sort of saying it, but we realize, all of our trialists, that we are really asking families and children to do a lot to go on these clinical trials. We know that they are pioneers, that this is not easy. And without their cooperation, we know we can't make progress. And I think anybody who's been doing this long enough feels a real honor to be in the field, but a real obligation to try to create a clinical trial that will be as least burdensome as possible on these families.
Truly.
Thank you so much for that comment, Dr. Packer. Unfortunately, that is all the time that we have for today. I just want to thank you all for joining us. And thank you once again to Dr. Vogelbaum, Dr. Packer, and Chris for your time, insight, and support of the brain tumor community.
Thank you.
Thank you all.
Aside from our webinar series, the ABTA has a variety of programs that help support patients and caregivers throughout their experience. For more information about ABTA's programs, events, and services, visit ABTA's website at abta.org, email us at info@abta.org, or call the ABTA Care Line at 1-800-886-2282.